Hello everybody, I'm Laura Piddock, Director of Scientific Affairs at the Global Antibiotic Research and Development Partnership, GARDP. I'm delighted to welcome you to today's webinar at the very beginning of World Antibiotic Awareness Week. Before we go into the details of our webinar, I'd like to just remind you of a few things about our webinar programme and Revive, which comprises it. So Revive is GARDP's educational and outreach activity where we aim to support the antimicrobial discovery, research and development community by connecting researchers, sharing knowledge and facilitating learning. Just as a reminder to everybody, all our webinars are recorded and they're available in perpetuity afterwards. So if you wish to re-listen to any of them, you may do so at your leisure and they're free. Please visit revive.gardp.org/webinars. Under our Revive programme, we also post antimicrobial viewpoints on topical subjects, including relationships or observations on antimicrobial resistance and COVID, or running a phase 3 trial in difficult circumstances during the pandemic this year, or even knowledge sharing about databases. And these can be found at the link shown here. We have also just launched our antimicrobial encyclopedia, and this is our latest resource that we're sharing with everybody, again free of charge, where you'll be able to find the common terms that are used in antimicrobials research, whether they be specific to R&D of discovery of new drugs, or perhaps just on antimicrobial resistance, so a term such as an MIC or those shown here. We will be adding to this all of the time, so if there's any feedback you'd like to give us, please do so. And if there's any terms that you think are missing, please let us know and we will do our best to add them. We're currently updating it about 10 terms a month at the moment. Thank you. So again, one final reminder to everybody today, if you're not familiar with attending our webinars, you can submit questions at any time during the presentations. You can do this on the right-hand side under the question section in your window, as shown on this slide. When all of the presentations are finished, I will ask the questions and our speakers will do their best to answer them within the time remaining in this webinar period. If we run out of time, then our speakers will be invited to answer the questions in writing and we will post them and you'll be able to see those answers below the webinar recording. So moving on to today's webinar, we have 4 speakers, starting with Jim Collins, and then we have Henriette, Willems and Fredrik Svensson and Cesar talking as well. So a really full programme where we hope to be able to share specific knowledge about artificial intelligence, otherwise known as computational chemoinformatics, to discover new antibacterials. So I'm going to first introduce Jim. I'm sure for many of you, he doesn't need much introduction. He's very well known for his research over the last several decades, I suspect. He is the Termeer Professor of Medical Engineering and Science and Professor of Biological Engineering at MIT in the United States. He's done an incredible amount of research and one of the founders of the field of synthetic biology, and he's focusing his work on next generation diagnostics and therapeutics. He's done a lot of work on pharmaceutical and medical devices and working with companies. And he's indeed helped to launch several companies himself. He's had numerous awards and honours, including a Rhodes Scholarship and a MacArthur Genius Award, and is a member of the National Academy of Sciences, amongst 2 other national academies in the US. So without further ado, I'd like to introduce Jim to do his presentation.
Thanks, Laura. Thanks firstly so much for the really kind introduction. Clearly identifying me as the oldest speaker in the symposium. But second, also, thanks for the invitation to speak today, and thanks to you and your colleagues for all the great work you're doing at Revive, and much needed work. What I want to do this morning is just share a little bit of our work on using artificial intelligence to get after the antibiotic resistance crisis. As all of us on the seminar webinar can appreciate, this is one of the existential crises facing humankind, and the problem's getting worse. We've been doing academic research for 3 decades, but really only became engaged around antibiotic work about a decade ago. And we got engaged really as a result of the data captured here, again, appreciated by many, which is that the number of resistant strains out there has been growing decade upon decade. This was a problem largely limited to hospital settings, but in the past decade in particular has now moved outside of hospitals into our communities, into childcare centers, sports fields, schools. And unfortunately, this crisis is being inadequately addressed by the commercial sector. The number of new antibiotics being approved and developed has been dropping decade upon decade, and unfortunately, the number of large biotech and pharma participating in the antibiotic space has been dropping, primarily from economic reasons. We got intrigued going back a decade ago, could we use network biology approaches to gain insight into how antibiotics act in order to figure out ways to either discover new ones or boost the effectiveness of existing ones. Starting now, just about a year and a half ago, I teamed up with Regina Barzilay and Tommy Giacola, 2 AI researchers here with me at MIT, to see if we could actually take an AI approach, specifically a deep learning approach to this problem. And what we did was join our efforts with grad students and postdocs in each of our groups, and the study was largely led by a star postdoc in my lab, John Stokes, where we decided to take an integrated experimental and computational approach to this challenge, really looking to unify machine intelligence with human intelligence. How we started out was we put together a very small training library. So this was an unfunded project. And the library consisted of 2,500 different molecules, 1,700 FDA-approved drugs, effectively including the known universe of antibiotics, plus 800 natural compounds. John applied these to E. coli and just looked to see which of the molecules had antibacterial activity as evidenced by growth inhibition. Given the very small numbers of these molecules in the training library, John discretized the data to just ask yes or identify the molecules as being yes, inhibitory antibacterial, assigning it a 1, or no, not antibacterial, assigning it a 0. We then took those data along with the structural information on each molecule and trained a deep neural net that effectively consisted of a message passaging neural net. This is a system that can look at each compound, atom by atom, bond by bond, and associate some phenotypic property with each of those atoms and bonds. And in this case, we had 2 phenotypic properties, antibacterial or not antibacterial. So we were able, through multiple training rounds, then train the deep neural net on these 2,500 molecules to identify which features of those compounds were associated with antibiotics and which features were not associated with antibiotics. We then took that model and applied it to the drug repurposing library that's available at the Broad Institute, where I also run a lab. And this consists of a little more than 6,100 molecules or compounds, many of which were in preclinical stage or even in clinical stage development. And we applied the model and asked the model to now identify molecules that are predicted to be good antibiotics. As an initial effort, shown on the left here, we went after and said, okay, let's take the top 99 unique predictions in the library to see which of those molecules actually exhibited antibacterial activity. We had 51.5% true positive rate, which is low relative to, say, an internet search, but decently high and useful from a drug screening ability or screening effort. We then went an important next step and said, okay, it seems like the model's got decent predictive capability. Now what we did was challenge the model to look again at the drug repurposing library and identify molecules that fit 3 criteria. 1, they needed to be predicted to be of an antibiotic with high prediction value. 2, of low toxicity using a differently trained model. And 3, didn't look like existing antibiotics, so had a low Tanimoto score. One molecule in that library of 6100 fit those three criteria, and that's a molecule shown here that we call halicin. We then decided to go in deeper analyses on halicin and study its possible antibacterial properties. And so John did a series of experiments looking for cytality against E. coli and found that halicin was highly potent as a bactericidal agent against E. coli, rapidly killing off different strains. We then looked to see if it was also bactericidal against tolerant E. coli. So these were E. coli that were in a metabolically dormant state or quasi-dormant state that were largely tolerant to most antibiotics. And just as an interesting aside, the initial effort we did for screening for our deep learning network was to see, could we identify compounds that were effective against tolerant bacteria? It turned out that there were very few hits in our training library. So amongst, say, the 300 antibiotics, only 6 had activity against tolerant E. coli. And amongst, say, the 50 biocides, only 7. So a very, very small number actually ever exhibited any activity. Halicin, however, as shown here, was remarkably bactericidal and against tolerant E. coli, making it as a quite exciting molecule. We then looked at 36 different multi-drug resistant, extensively drug resistant, and pan-resistant bacterial strains from the CDC and found that halicin was effective against 35 of those 36, with the exception being the Pseudomonas strain. We then went and did in vitro work against TB, and again, excitedly found that halicin, molecule identified with our deep neural net model, was actually quite bactericidal and rapidly bactericidal against TB. And these are just in vitro work here, but we subsequently have shown that halicin also is effective against macrophage-engulfed TB. We then wanted to see the resistance background, both to identify potential targets and mechanisms of action, but also to see what was the resistance spectrum for halicin. Compared it to Cipro, and as shown here on the left, within a small number of days, we saw resistance grow against Cipro, and 30 days out, we had resistance that was several hundredfold higher against baseline. For halicin, we didn't see any resistance after a few days, and remarkably, after 30 days, we did not see resistance. Of course, if we look long enough out, we'll see resistance, but it was, again, quite encouraging on this initial set of experiments. John then dug in to look at the way identify the target or mechanism of action of halicin, and it doesn't appear to have a specific molecular target. But on the basis of RNA-seq analyses and phenotypic follow-up, John was able to identify that halicin appears to act on the membrane, selectively dissipating pH component of the proton motor force, disrupting the ability of the bacteria to generate energy. We then wanted to see, could halicin have efficacy not simply in a dish, but also in various in vivo animal models. And so we set up a C. difficile infection. So in this case, we're going to deliver halicin systemically. And C. diff, as you know, is a gut pathogen that's highly problematic. In the US alone, it can account for about 250,000 infections, resistant infections each year, resulting in 13,000 to 14,000 deaths. It does not typically respond to our antibiotic arsenal, and in the most extreme case, requires a fecal microbiome transplant. We found, however, in these animal experiments that halicin was actually really quite effective at eradicating C. diff infections. We then set up a second animal model, and this was for Acinetobacter baumannii. As many of you know, Acinetobacter baumannii is a nasty pathogen. It's at the top of the list of the WHO's list of nasty pathogens for which we desperately need antibiotics. It's also known as the Iraqi bug, and particularly US and UK soldiers in Iraq and Afghanistan have often pick up very nasty Acinetobacter baumannii infections in skin wounds that are very difficult to treat. In this skin infection model, John actually found that halicin was remarkably effective again at treating the Acinetobacter baumannii infection model. We then wanted to see, could we extend the model to a much larger library? So we looked at the zinc database. The zinc database is a compound library consisting of 1.5 billion molecules. It would be impossible to empirically screen such a large library. But with an AI-based model, a deep learning model, it'd be quite straightforward. we decided to focus on about 10% of the library, so about 107 million molecules, screening it through with our deep learning training model. It only took 3 to 4 days to screen such a large number of molecules. Not expected, or as expected, we identified several hundred to be predicted to be very high antibiotics, and again went in and said, okay, can we identify those that are predicted to be good antibiotics but don't look like existing antibiotics? We identified 23 such molecules that we could either synthesize or purchase. We brought them in the lab, tested them, and 8 of those exhibited antibacterial activity. 2 of which, shown here, had actually broad spectrum bactericidal activity against a wide range of bacterial pathogens. Where are we going with this? Well, we're taking this platform in many different directions. With John, with Regina, with Tommy, we've launched the Antibiotics AI project at MIT with the audacious goal of using AI to discover or design a number of novel classes of antibiotics over the coming few years. We're taking the platform in many different directions, so we're collecting now larger training library data sets. We've now expanded our training set to 40,000 compounds. We're going to go beyond, or going beyond E. coli to 7 different pathogens to both look at broad spectrum and narrow spectrum antibiotics. We are also expanding the platform to get after target prediction and mode of action prediction, as well as a prediction of combination approaches. We, moreover, are using the platform now not only as a screening tool against libraries, but also as a design tool. So opening up the machine learning models to see what was learned from a chemical structure standpoint and using those data to design and synthesize novel molecules that can make for good antibiotics. As a related project, speaking to identifying mode of action with Jason Yang, star postdoc who's in my lab, who's now a professor at Rutgers, Jason actually developed a platform to get after white box machine learning for antibiotic discovery. And in this case, what he was able to do was collect training data on perturbations to, in this case, E. coli, and train a deep model, a machine learning model that was based on the underlying metabolic network of the pathogen, and was able to identify that the antibiotic action led to adenine limitation that looked to have purine biosynthesis in response that led to ATP demand, and this contributed significantly to antibiotic stress that led to cytality. With that, I want to thank the team of young stars whose work I got to share with you today, and want to again thank Laura for this invitation. And with this slide, I want to pass it back to Laura to introduce the next speaker.
Thank you very much, Jim. So we'll now move on to speaker number 2 of our 4 speakers, and this is Cesar. He is a presidential assistant professor at the University of Pennsylvania and leads a machine biology group to integrate synthetic biology, microbiology, and artificial intelligence, and aims to also use computer-made tools and medicines to replenish our current antibiotic arsenal. Cesar was recognized by MIT Technology Review in 2019 as one of the world's top innovators and was selected as the inaugural recipient of the Langer Prize in 2019. And this year, the ACS Infectious Diseases Young Investigator Award. He's also been named as 2018 Wunderkind by Stat News, a top 10 under 40 in 2019 by GN, and is one of the 30 rising leaders in life sciences. And he's got numerous articles, as indicated here, and has secured multiple patents. So again, we have a leader in the field. So thank you very much, Cesar. Over to you.
Thank you, Laura, for the very kind introduction. And thank you for the invitation to share some of our recent work in trying to use computational tools to try to come up with novel antibiotics. So the motivation, as Jim mentioned in the previous talk, is pretty obvious. We're heading towards a world where 10 million people are projected to die as a result of drug-resistant infections, which will actually surpass every other major cause of death in our society, including cancer, as we can see here on the slide. And those 10 million deaths actually correspond to one death every 3 seconds. So we're really heading towards a post-antibiotic era, unless we do something about it. And let me just remind you that, of course, antibiotics are the foundation of modern medicine. Without them, we could easily die from infections stemming from injuries and childhood maladies. And they, obviously, without them, surgeries, transplants, childbirth, and chemotherapy would not be possible. And so really, the vision that we have in my lab about this is that although the natural world has been a great inspirator, source of inspiration of antibiotics over many, many years since the discovery of penicillin in 1928, for decades, we have been unable to discover a truly novel class of antibiotics in the natural world. And bacteria have, of course, become increasingly resistant to existing drugs. And so our hypothesis is that perhaps the next generation of antibiotics is likely to come not from nature, but instead from machines. So what we're trying to do is we're trying to encode all these chemistries that have evolved in the natural world into ones and zeros, so computers can take care of the discovery process. And more specifically, if you go through the tree of life and you look for molecules with intrinsic antimicrobial properties, you will find this family of small proteins called host defense peptides, or HTPs for short. They're very small, between 10 and 50 amino acids in length. They're, of course, genetically encodable because they're peptides, and they're chemically synthesizable. So they're pretty easy to manipulate in the laboratory. And they're highly conserved, like I mentioned. Bacteria produce these molecules, and we produce them as part of our immune system to help us fight off invading pathogens. So we've centered a lot of our efforts in trying to engineer these molecules by using very simple rules from protein engineering that tell us that if you can control the sequence of amino acids that make up these molecules, you'll be able to control their biological function. And so I liken this to playing Lego in the laboratory. That's basically what we do. We try to rearrange the building blocks of these molecules into specific positions to create, in this case, novel functional antibiotic molecules. And the framework that we use in the laboratory is we basically want to understand these molecules from first principles, and we've devoted a lot of our time to doing so. Once we know that, we can control them in the laboratory, both chemically and biologically. And finally, once we've reached that level of control, we're in a good position to encode or teach all that information onto computers. And that's what I'm going to focus my talk on today. So one of the things that we did is we took some of these natural molecules and we trained computers to execute Darwin's algorithm of evolution to be able to evolve them to become better antibiotics. The reason why we use Darwin as an inspiration here is that we thought that what better way to optimize or innovate a molecule than to use a process that has been happening for billions of years, and that's, of course, the evolutionary process. And so what we did is we took, as an initial population, we took these HTPs from the natural world, and then we trained the computer to execute a sequence of events, evolutionary events, consisting on mutation, selection, recombination, and then n-fold iterations of that process to create innovation. And so on the right here, you can see how over different iterations of the optimization process, we can see that the molecules go towards an optimal fitness value that correlates with predicted antibiotic activity. So we're basically training the machine to turn molecules that are not so good into optimized versions. And in the process of doing so, interestingly, we can explore previously unexplored regions of sequence space of these peptides, and yielding molecules with sequences that are unlike what we typically see in the natural world. So here I'm showing some of the peptides the computer generated. These are computational simulations. We can see that they form these alpha helical structures. But then we reach a roadblock that is common to any kind of work when you're doing computational studies, and that everything that we have here is just a prediction. We still need to experimentally validate that, and so that's what we did. We synthesized a number of these molecules, and then we tested them against actual multi-drug resistant bacteria, and we found a lead molecule that had a very low MIC compared particularly compared to the templates that the computer started the simulation with, which were essentially non-antimicrobial or not very good antimicrobials. And we did this on purpose to demonstrate as a proof of principle that we could train a machine to turn a non-active molecule into an active one. Here I'm showing, this is an NMR structure of the lead molecule. Again, we can see that beautiful alpha helical structure. And again, that's the same thing, the same molecule. And of course, we're always interested in translating everything we do in the lab into real world applications. And our overarching goal is to generate new antibiotics using computation. And so we tested our computer-made molecule in the context of a mouse infection model. And interestingly, the machine-made antibiotic was capable of resolving the infection better than 2 of the templates that we started the optimization process with. And then on the left here, we have the untreated control group of mice, so you get a reference infection load from the untreated group. So in addition to using computers or computational tools to design new molecules like I've just described, we're also interested in using computation to discover novel antibiotics, perhaps in places where people have not traditionally looked before. And there's a lot of those places if you start looking into genomes and proteomes. And to address this problem, we decided to take inspiration from image and speech recognition algorithms, but instead of recognizing facial expressions or sounds, we wanted to recognize amino acid patterns, in this case, that constitute antibiotic-like molecules. And because of our previous work, we knew what to look for. And so it was relatively easy to build an algorithm that could look for those molecules, say, in genomes or proteomes. It's analogous to having a large Word document where you're trying to find the word hello, so you know what you're looking for. And you go to the search function and you find the word hello. So in this case, we knew what the word hello was, we knew what we were looking for, and so it makes it a lot easier to actually find that within a huge data set. So what this allows us to do, just for reference, is if you have an entire protein in 3 dimensions, you can display it also in 2 dimensions. And the algorithm, what it does is it runs through the code, in this case the amino acid code of the protein, and it identifies regions within that code that represent predicted antimicrobial hotspots. And those are represented here by the different colors. And this is given by a particular score that we developed that is called an antimicrobial score, or AS for short. Again, this is only a computational prediction, but in this case, it's a very powerful one because it allows us to identify regions within entire proteins, for example, that we can then actually extract from the native protein, and we can play around with these molecules in the laboratory. So we've applied these algorithms to try to mine the entire human body for novel antibiotics. And interestingly, we found antibiotics, not only antibiotic molecules encrypted, not only in the immune system, which is where you would imagine these molecules to be, but also in other systems throughout the body, including the nervous system, cardiovascular, and digestive systems. So we're now coming up with all these hypotheses that this is a way for linking the innate immune system with other systems throughout the body to have a more holistic, sort of connected approach or system that connects different systems throughout the body. And also, it may be a way for preserving energy at the genomic level, where instead of having one gene encode for just one protein with one function, you have one gene encoding for one protein with multiple functions. So we've taken a number of those molecules from the human body, and another interesting thing that we found is that if you take the molecules produced within a similar biogeographical area within the body, and you combine them, they synergize, they potentiate each other's activity against pathogens to low nanomolar range, which was very interesting to see. Also, interestingly, bacteria appear to have a hard time developing resistance to these encrypted antibiotics, in this case over 30 days, the period of the experiment. And as a control here, we have polymyxin B, which is obviously it's a conventional antibiotic that bacteria eventually develop resistance to this agent. And then we took some of these encrypted molecules, again, that we identified using the pattern recognition algorithm, and we tested them for anti-infective activity in a mouse model. And we found that not only in monotherapy, but also in combination therapy, again, thanks to their synergistic or potentiating effect, when you combine different ones, you get an enhanced antimicrobial output. We can also use these pattern recognition algorithms to as I mentioned, browse or mine other information. And we have a side project now where we're looking into extinct organisms, including the woolly mammoth, to try to find these molecules in their genome and the partial genome that is available, and also the proteome. And finally, I'd just like to mention that we've been involved in a number of projects now trying to exploit venoms as a source of novel antibiotics. And in this recent paper that we published a couple of weeks ago, we used a computational approach to first find a motif in a library of peptides that was associated with increased antimicrobial and immunomodulatory activity. And that, then we engineered that motif into one of these venoms. And we were able to show that the synthetic derivatives had that dual activity of being able to resolve the infection by directly targeting the microbe by perturbating its membrane, and also resolving the infection indirectly by boosting the immune system, and at the same time dampening the inflammatory response. And we've shown this in vitro and in animal models. And in this case, we also demonstrated efficacy of this dual antimicrobial in a sepsis infection model. And again, sepsis is incredibly important. It kills 11 million people a year, and which corresponds to about 20% of all deaths in the world today. We demonstrated that both in laboratory strains and also in clinically relevant multi-drug resistant bacteria. And with that, I'd like to thank the team that made all this work possible. It's really a privilege to work alongside them, and thank our funders who also obviously made everything possible. And here on the bottom left, you have my contact information in case you want to reach out. And thanks for your attention, and I'll hand it over back to Laura.
Thank you very much, Cesar, for another excellent presentation. So the last part of the presentations in this webinar are going to be delivered by Henriette Willems and Fredrik Svensson. So Henriette is a Senior Research Associate at the Alborada Drug Discovery Institute at the University of Cambridge in the UK. She obtained her PhD in 1996 from that university, and then she worked for several small biotechnological and large pharmaceutical companies, including Astex Pharmaceuticals, GSK, and Takeda. Henriette has published on several structure-based drug design projects and contributed to drug discovery patents in a wide range of therapeutic areas. Fredrik Svensson is a Senior Research Associate at the Alzheimer's Research UK, University College London Drug Discovery Institute in the UK. He was awarded his PhD in 2015 from Uppsala University in Sweden, after which he undertook a postdoctoral fellowship in chemoinformatics at the University of Cambridge with Andreas Bender. Fredrik has published extensively on the use of machine learning in drug discovery. Thank you, Henriette and Fredrik, for your presentations.
Thank you, Laura, for a very kind introduction. I'll be talking to you today about computational chemistry on a budget. So we're changing tack a bit with this presentation and focusing more on methods and tools. First of all, I thought I'd tell you a little bit about myself and Fredrik and where we work. So we both work for the Alzheimer's Research UK Drug Discovery Alliance. So that's funded by Alzheimer's Research UK, and so we work on dementia targets. So sadly, we're not experts on antibiotics, but I hope our knowledge can still help you. There are three different institutes, three sister institutes. I work in the Cambridge Institute, Alborada DDI. Fredrik works at the UCL Institute, and we also have an institute in Oxford, where sadly at the moment we don't have a computational chemist. Both Fredrik and myself work primarily in the drug design part of the unit. We also have chemists to do synthesis. We can do screening and take compounds all the way to in vivo proof of concept. So the reason we are talking today really is because of a recent paper where we discussed how you can do computational chemistry on a small budget. And so if there's anything that catches your interest in this presentation, you can find more information in this paper. What we'll be talking about today is how computational methods can help in target identification, and that's looking at expression, biomarkers, finding known compounds, how methods can help in drug discovery, so for example virtual screening, library design and property prediction, and how computational chemistry can help in hit-to-lead methods. So moving on to target identification. Target identification can be a huge task because there's a lot of information out there, and finding it and putting it all together can be very time consuming. And so our colleague who used to work at the Oxford DDI, Stefan Lecesco, wrote this program called GARDP, which you can freely download from GitHub, and it's also in this paper. And this tool can take a gene name or multiple gene names and go off and find for you known literature, known ligands, crystal structures, tissue distribution, disease association, etc. It will search all of the databases that you can see in the central panel, use some SQL codes to put that nicely into an Excel spreadsheet with a tab for each database. And then finally, it uses some machine learning to then predict whether your target is a tractable target for drug discovery or whether it may be very challenging. So that's a very quick and easy way for us to do some quick analysis of targets and also to compare different targets and decide which one may be the most appropriate. Moving on now to hit discovery. Traditionally, sort of hit discovery is done by high throughput screening. And this is great if you're in a big pharma company, you have a large library of molecules you can screen and you have the budget to do that. But if you're on a much smaller budget, what can you do? So options are a focused library screen where you can maybe purchase from a vendor a library more targeted to what you're interested in. You could do iterative screening where you screen only part of a library and use the information you gain to then decide which compounds you have to screen next. So it still requires a large library, but you may save on the consumables. Or finally, you can do virtual screening, and that really saves you a significant amount of cost and time. Whichever way you go with screening libraries, it always pays off to ensure that you have good quality compounds to screen. So we always start off by applying reactivity filters. So these are filters that will pick out the molecules that are likely just not to be stable in the solution or just even in a vial. We screen for paints. These are things that will interfere with the assay, so maybe they're fluorescent or maybe they're aggregators. They do come in many different guises. There are about 400 different paint filters, and some of them are more appropriate than others depending on your target. And both of those are referenced at the bottom of this slide if you want to find more information. And then we tend to filter by property. So for us, that's mainly about making sure compounds go through the blood-brain barrier, but it could also be about solubility, or maybe you have specific filters for things to get into the bacterial cell. There are many tools available for this that are free, or at least free to academics. KNIME and RDKit are completely free, Chemaxon free to academics, so many ways to do this very cheaply. Then moving on to virtual screening. So you can go two different ways with virtual screening. You can use a ligand-based method or a receptor-based method. So if you only have known active ligands, you can use that to build a model, maybe depending on the shape and the electrostatics of the ligand you have, or you could build a pharmacophore that focuses on shared features of the molecules that you already know about. Or if you have a crystal structure or maybe a cryo-EM structure, you could go for a receptor-based method. So the best known of that is docking, which you can now do really fast, and so you can screen a lot of compounds that way. Or you can use a receptor-based pharmacophore, where you just pick out key features and the shape of a protein. In either case, you'd need a library to screen, and this is typically a library of compounds you can purchase from a vendor, which you then convert into 3D digital molecules for screening. Now, once you've decided which types of virtual screening you want to do, it's still worth considering how likely your virtual screen is to be successful. Because, for example, if you have a crystal structure, you're in a good place to start a virtual screen, but all crystal structures are not equal. Some are missing electron density, so always check for density in the active sites, or missing residues, partly missing residues in the active site. For example, lysines quite often are modeled in the PDB file, but when you look in the density, actually the side chain just isn't there because they're flexible and move around. And if you have multiple crystal structures, you could look at changes in the conformation that might help you improve your model. Then it always pays off to try and find all known inhibitors and also compounds that are known perhaps to be inactive for your target. And then you can use that to validate your protocol, dock these in or fit them to your model and see whether it can distinguish the active from the inactive. Now, once you've decided on your virtual screening protocol, you need to decide which database to screen, and it pays off to think about this carefully. When we think about success in virtual screening in our unit, we would like to see 10 to 50 small hits in small clusters, ideally with some SAR, and small clusters means different types of molecules. Because we need to sort of account for some dropout, maybe because of flat SAR or intractable chemistry, and so on. We typically get a hit rate in a virtual screen of about 1% if we call anything better than 10 micromolar a hit. And that more or less fits with what is reported in literature, although there is a wide variety of studies. HCS screens have a much lower hit rate, so you're looking at 0.05% at a 10 micromolar cutoff, but of course it depends on the target. So that means that for a virtual screen, you would need to purchase and actually screen about 1,000 compounds to find your 10 to 50 hits. And for a high throughput screen where you just have a random library, you'd need to screen about 20,000 to get your 10 hits. And that is associated with a certain amount of cost. So it pays off to sort of think about how many compounds you want to buy and where you might find compounds that you can afford. So single source databases such as Bioascend, ChemDiff and Enamine tend to be cheaper than those from aggregate vendors such as Molports and eMolecules, which are brilliant for lots of choice if you know what you want, but do tend to work out a little bit more expensive. So just an example of a virtual screening workflow that we use is in this project. And we started off with 1.5 million compounds from a vendor library. We did our property and substructure filter, took at reactive groups and paints and so on, and came out with 500,000 compounds that we liked. Then we protonated those and generated stereoisomers using ChemExon tools, which again are free for academics, and converted them into 3D molecules using RD kits. Again, this is a completely free program. We did a quick pre-docking and then a pharmacophore screen to select those compounds with the best features. and then redock those and selected the top 1,500 compounds by 3 different scoring methods. And then finally, we like to cluster our compounds to get some diversity in the compounds we finally purchase. So we clustered using Data Warrior, which again is a free program, and ended up buying 850 hits, and then got 12 hits from that at less than 10 micromolar. So that worked out very nicely for us. And just to compare how a virtual screening stacks up against high throughput screening, we did 2 virtual screens for this project that each costs just a few £1,000, one £2,000, one £4,000 for compounds and screening. Hit rate around about 1%. We got 11 hits from one and 8 from the other, and the total time between 7 and 8 weeks for the virtual screens. The high throughput screen, on the other hand, costs 10-fold more. Hit rate obviously lower, but you do get more hits because it was a much larger library. Not necessarily more diversity in those hits. And it did take quite a long time in the screening. So overall, we'd say virtual screening, cheaper and faster, and can work very well, but you do get more hits with high throughput screening. At this point, I would like to hand over to Fredrik, who's going to talk you through the hit to leads.
Thank you, Henriette. Yeah, so I'll talk a little bit about computational methods in the later stages. And a typical thing you'd like to do is starting from a hit, or in this case, a literature compound. You'd like to optimize something with this compound. In this case, we wanted to make this brain penetrant. And the sort of strategy we adopted is a structure-based design using the kind of methods Henriette already introduced, as well as prediction of various properties that we think are important for the target. And in this case, you can see we managed to preserve activity and increase the brain to plasma ratio quite significantly, maybe not quite to the level we'd like, but still tracking properties can be very important for these kind of studies. And typically this would be done using either matched molecular pairs or a QSAR or QSPR model, so a quantitative structure activity or property relationship model. And the idea is to prioritize what compounds to make, because typically you can make more compounds than you actually have, you know, the choice, you have more choice than what you can actually make, and you need to narrow that down somehow. And these kind of methods can be very helpful for that. And there's a lot of freely available data in various databases that you can use for this. And especially for properties such as solubility or various ADME properties or log P, these that can be transferred between projects, you can then sort of build up your data as you progress between projects. So in matched molecular pairs, you identify small chemical changes between molecules and an associated change in property. And by looking in a large databases of such changes, if you want to improve a particular property, you can see what kind of change historically have improved that property. Whereas a QSAR model is typically a machine learning model that you'll train on a lot of data. Just a quick summary of what we can and cannot do with these kind of methods. So doing virtual screen, molecule confirmation, searching databases, doing basic property predictions, these are all kind of straightforward things. It gets more difficult when more flexibility is included in the protein structure, or very challenging when you want to predict binding free energies or large-scale protein movements, or anything that involves a more involved biological process with, for example, multiple mechanisms. A few words on the computational environment required to do these kind of calculations. And first, something that is often forgotten, and that is making sure that your data is managed. Because if you want to use your data in the future to build predictive models, for instance, you need to make sure that the data you generate today is actually stored somehow. And there are commercial solutions for this, but you can have any solution, but just making sure that data is managed and that metadata associated with that data is stored is really important. In terms of a computational environment, what we found work best is to have a mix with sort of a basis around the commercial software suite, and then complement that with various open tools and workflows. And this also future-proofs it so that our commercial software suite will have a lot of documentation and things like that. So your computational chemist would move on, someone else can quite easily take it over. And in terms of hardware, these kind of calculations require only relatively modest hardware. So with a modern desktop machine, you can run all of these calculations that we discussed here today. It might be useful to also have a server. While discussing software, just very quickly like to issue a caution around some of these web pages or online tools that allow you to do some of these calculations, which can be really handy, but think twice whether or not you're comfortable actually uploading your compounds to somewhere where you don't have control over what's happening to them. So finally, like relatively small resources can go a long way in supporting drug discovery. So essentially what you need to do these kind of calculations is a computational chemist or someone to do the calculations, some modest hardware, which is not a big cost and typically a one-time cost, and then some software, and the costs here would be approximate sort of ballpark figures for annual license costs for a commercial software suite, depending on if you're an academic or industry. Obviously, this depends on your exact requirements and how many users, etc., but just a ballpark figure what you need. And with this kind of investment, I hope we've sort of given you an introduction to that you can deliver value to the drug discovery process by faster evaluating targets, doing more cost-effective screening, and essentially getting better compound quality quicker. And with that, we'd like to thank all of our colleagues at the Alzheimer's Research UK Drug Discovery Alliance. I'll hand back to Laura for the Q&A session.
Thank you very much. So we've received quite a few questions already, but those of you who are listening hard and have not yet had time to submit your questions, please do so now. As you can see here, there's a reminder of how to ask your question on the panel on the right-hand side. And we will do our best to answer all your questions now. But if we do run out of time, our speakers will be invited to respond in writing, and we will post these answers with the recording of the webinar in due course. So I'm now going to move to the question and answer session, start with some of the questions that we've had. So, oh gosh, we're getting quite a lot. So I think we'll start with the with our last speakers and work backwards, if I may. So for Henriette and Fredrik, how much data do you need to build a QSAR or MMAP model?
Yeah, so ideally to build a predictive model, if you want to build a QSAR model, you'd need maybe 100 or more compounds, but you can use methods to start to look at trends already when you start talking about tens, but to build a proper model, it would be in the hundreds or even more for a really good sort of general model that you use across projects, trained with data from Campbell. You have thousands of data points ideally.
Thank you. So we're getting a couple of questions now about the types of approaches that you have taken, all of the speakers, when I say you, so using a variety of different types of algorithms and searches. And one of the questions I've had, and perhaps Henriette, you could answer this, is can these approaches be used in resource limited settings, in particular in low or medium income countries?
Yes, I think so. I think there are a lot of free tools out there. Some of the ones that we already mentioned are NIME and RDKit, which is associated with it, that has a lot of computational chemistry tools, including ways of sort of loading in molecules, calculating properties. You can do machine learning, etc. So that is really quite a nice package for lots of different things, and has very good documentation online to help you get started. There are several others, and again, in the paper we published, we have a big list of softwares and which ones are free and what they can do.
Thank you. In fact, several people have noted that some of the softwares and certainly one of the data storing platforms that you mentioned are quite expensive. I'm reading two questions at the same time, so do forgive me, but they're basically saying the same thing. So what they wondered is, can the speakers after this indicate the free resources available, which would not necessarily be very expensive packages? And secondly, what other database platforms are there other than the two that were mentioned in the last presentation? So I don't know, Fredrik, you'd like to answer that?
Yeah, I mean, essentially, those tools are just the pre-made and quite user-friendly databases that you can use to store your data. But a lot of people have homemade versions of it where you can use any database tool, like MySQL or something else, and set up your own data server. Or you could have something even more primitive than that. You could have just making sure that you store your Excel spreadsheets in a good shared folder somewhere that everyone have access to, and that you store associated metadata. So not just an Excel spreadsheet, and 5 years later you go back and say, what did I actually have in this spreadsheet? You have no idea anymore. But some sort of documentation, what you've stored where, can go a long way compared to having nothing.
Thank you. And then still on technology, but really picking up on the zinc database, a very large compound database, which both I think Henriette you mentioned and you as well, Jim. Can these be used with very limited resources?
Do you want me to go first there? I would say if you want to do docking, that's hard because you really need a large computer system, a cluster ideally, to do docking of such a large database. However, methods like the ones we saw where you do more 2D picking out of molecules, even 1D, so just really on the molecule strings, they are much faster, and that's much more feasible. So you could do similarity searches, decide which molecules you're interested in, and then just focus on that smaller set perhaps to do docking.
Thank you. Question now about TargetDB, which you mentioned, because clearly your research is not on antimicrobials, whereas many of our target audience do work on antimicrobials. So the question is, can TargetDB be used for microbial targets?
Yes, it has all of the protein targets, I believe, that are in the various databases. So assuming that the underlying databases, which you briefly saw on the slide, have the antimicrobial targets in, then they will come out and you can find the information using TargetDB.
Thank you. So just looking, I've got color coding now, so you have to bear with me for each of the speakers. So so let's have a look here. So Jim, do you have an assessment of the applicability domain of your deep learning antibacterial model? And if so, how you use it? Do you believe the model is sufficiently global to predict activities of any type of molecule?
So I believe that the platform that we used is not specific to antibiotics, meaning the underlying architecture and the methodology, the pipeline methodology, is independent of antibiotics. The data we collected was specific to E. coli, which was also intriguing of some of the predictions we got for molecules that had activity outside of E. coli. But the actual platform of screening for phenotypic data, having compound structure information, training a deep neural net, and then using that for either screening or design, can be extended to other clinical indications. For example, as part of the AI Cures effort at MIT, we've been using the platform to look for antivirals against SARS-CoV-2.
Thank you. And indeed, the next question linked with that one was, are you considering using the same approach for antifungals to identify potential antifungals?
Yeah, we are considering it. It's actually, interestingly, probably the most common question we're asked for extension, not into cancer or diabetes or even into SARS-CoV-2, it's antifungals. And we've done some work around antifungals in my lab, not extensively, but we now are exploring what kind of training data could we get that, for example, might enable us to screen for molecules that could be effective against C. aunins.
So we've just talked about different microbes. If we're back with bacteria, this question is saying, does your model take bacterial species into account, gram-positive versus gram-negative, and can identify potential hits for either of those groups of bacteria?
So the underlying model does not actually have, as we currently constructed, any particular biological feature that would be specific to a bacterium versus another cell, or different types of bacterium, say gram-negative versus gram-positive. It's all very much a function of the training data. So the only data that we've used extensively so far was on E. coli. But we have efforts now underway looking at staph to expand to gram-positive. And as I mentioned, one of our plans as part of the audacious Antibiotics AI project is to collect data across many different species, both pathogen and commensal, with the goal of training models that would allow us to identify or design narrow spectrum antibiotics.
So your model is iterative, in other words, every time you learn something new, you build it back in and refine the model.
Yes and no. So the model is iterative in that you can add additional features. I should say iterative modeling and machine learning is a contemporary hot topic. In most of these cases, we're actually training completely new and independent models off of the data set. So you often just go back to the starting point. It doesn't take very long. You can do it over a matter of a small number of days, but it does become iterative from the development of new improved models with the new data sets that you get.
Thank you. Moving on to Cesar, we have several questions along a theme about deriving molecules from molecules that are used in the host immune response. So this is the first. So when giving different segments of mammalian proteins as potential anti-infectives, do you see toxicity or adverse effects on the immune system?
Yeah, that's a great question. So we have tested the encrypted molecules. Again, the way they work is essentially you have an entire protein sequence, and they're predicted to be cleaved off, this segment. So the segment gets cleaved off, and that's the encrypted molecule that we're finding. And we've tested the encrypted molecules in a number of mouse models, and we haven't seen any major adverse effects. We haven't yet tested specifically. We haven't done immunomodulatory assays to see if they have some sort of immune boosting effect or adverse effect against the immune system. But so far, with the experiments that we've done, we haven't seen anything that would indicate that they would be toxic. And conceptually, I would be surprised if they were, because they are derived from our own bodies. But it's work that we still need to do.
And related to that question, we have another one saying, could these FDPs somehow disturb the immune system or its regulation?
Yeah, sure. So a lot of the HDPs are known to have immunomodulatory properties intrinsically, their natural sequence. So, for example, they can stimulate, they can balance different production of different cytokines. Some of them, like I mentioned with the venom-derived molecule, you can get effects that have sort of anti-inflammatory properties, which may be interesting, right? Whenever you have an infection, you have inflammation, which you need to resolve the infection, but that process can become too exacerbated at times, which can lead to sepsis, which is obviously a huge adverse phenomenon. And so being able to boost the immune system in a good way and then balance that inflammatory response is interesting. And some of these molecules have some of those properties. And now what we're trying to do in the laboratories is try to understand what are the sequence requirements that make those molecules have those properties, and then try to potentiate them, right?
Thank you. Using the approach that you have, Cesar, how do you determine the ideal length of a peptide antibiotic? Yeah,
that's a good question. So we're trying to simplify things so far, and we're reducing our search to small molecules. Typically, the threshold is what is the maximum length that is chemically synthesizable, that we can then synthesize in the lab and validate the computational models. And that's around 50 amino acids. So we typically reduce it to that. It's very difficult with solid phase synthesis. Chemical synthesis is very difficult to synthesize anything beyond anything larger than that. So that's, we keep it, we're pretty practical in that sense.
Thank you. Another one for you, Cesar. How do you deal with the usual issues? I say usual, those that people consider usual for peptides, such as penetration and stability.
Yeah, sure. So with basically all the computational work that we've done so far, we're focusing on linear sequences, which have some issues oftentimes with, like you mentioned, they can be degraded by proteases, etc. So what we do is downstream, we have the capability of chemically modifying those structures to either cyclize them or incorporate non-canonical amino acids. We have a lot of experience incorporating the enantiomeric versions, for example, which is basically the mirror image of the amino acid, which is not recognized by proteases that would degrade it. So at the moment we're doing that downstream. It is very difficult to encode computationally non-canonical amino acids. It's something that we're thinking about at the moment, but we haven't done it successfully yet. And the same applies to cyclic structures or constrained structures that would have more desirable stability profiles.
Thank you. This is a question for Jim now. The true positive rate for 100 compounds from which halicin came from was 50%. Do you know what the rate would be if you chose 100 compounds at random, i.e. a negative control for your machine learning technique?
Yeah, we did that experiment. We found it to be about 2 to 3%.
Thank you. So the next question, again, another one for you, Jim. How do you incorporate microbial attributes into the model? I think they mean antimicrobial attributes.
So I'll maybe answer it both ways, Laura. So the antimicrobial attributes will need to be quantified either discretely, 0, 1, or in analog fashion. And ChemProp, the underlying system we use with the message passing neural net, can accommodate a good number of such ones that would be done in sequence, albeit building a single model. The second, say not getting after per se antibacterial, but actual bacterial properties, speaks to the very last slide, which was Jason Yang's work on white box machine learning. I do think there's a lot of potential in this space, both for basic biology as well as applied biology, wherein many of the models we use in machine learning, if not the great majority, are black box and thus not particularly insightful from a mechanistic standpoint. And there, where one can do is don't just develop the model as one traditionally would as a deep neural net, but instead train a model on an existing or extant biological model. So for example, Jason did it across a metabolic network. One can envision doing that across a pathway-based model and or a transcription and regulatory network model.
Thank you. This next question is for Fredrik. Do you have the KNIME workflows and others shared in your publication or via GitHub?
Well, we didn't share any specific KNIME workflows in our publication, but there is a large database of various workflows on the KNIME webpage that are freely accessible, that anyone can go in and just search and download.
Thank you. And a linked question to that is, are these workflows accessible to those working on antimicrobials? They didn't say any specific species or type of microbe, but would they be applicable?
I mean, I obviously don't know if there are any specific workflows. There probably are specific workflows that someone designed and put out there. but there are also, I mean, there are a lot of general tools that are helpful for drug discovery, regardless of what area you're working in. So there's definitely applicable tools.
Thank you. This is one for Henriette. Can you please expand about how you would proceed to do virtual screening without a 3D structure or model of the target protein?
Yes, so that would be ligand-based virtual screening. And there are two methods for that, really. One is obviously you need some known ligands, otherwise it is nigh on impossible. So first thing to do would be to go and find your known ligands. And then there are either tools available, and there are some free tools and some commercial tools, to superimpose your molecules to try and find features in common, and then you can make a pharmacophore. Or alternatively, you could just take a single molecule and look at the shape. And for example, a tool like ROX, which is a commercial tool, would then sort of screen a database based on just the shape of your molecule. Again, there are also free academic tools available who can do this sort of thing. Yes, again, sort of our paper contains some references. You can just Google it. There are definitely a number of tools out there for doing this work.
Thank you. This is a question for Jim. Your hits from the zinc database had 1 or 2 nitro groups per compound. Was this selected from your AI algorithm or simple phenotypic screening?
So the molecules that we identified and pursued were identified by the AI model. So it wasn't from, the phenotypic screening was used to train the model, and then the model was applied to the different databases. You know, the halicin itself actually has a nitro group, so it's similar to antiparasitic compounds, and its closest antibiotic from a Tanimoto score is metronidazole, which has a nitro group, although the Tanimoto score was 0.2. So the model seems to like nitro groups, and as many know, nitro groups can be problematic for many compounds, although not universally so. Our initial work on halicin indicates that it has low tox profiling. I made reference to a study that we're about halfway through that I'll be excited to share with the community in the coming few months, which is opening up the trained models to really identify what molecular substructures and scaffolds are being enriched for, and how do those change when you're training the model with known antibiotics, and how does it change when you train the model leaving out the known antibiotics, and in what way can we bias the system toward discovering completely new scaffolds. So there's, I think, much to be uncovered. But as the listener asked, yeah, it was interesting to see some enrichment for nitro groups in some of the compounds that we found good hits for.
Thank you. This next question is for Cesar. Can you predict peptide stability in vivo?
That's an interesting question. So we are trying to do that. So for example, in the genetic algorithm that I showed, and circling back to some of the previous questions, actually, you can incorporate descriptors in the fitness function that select either against gram negatives versus gram positives. So you can differentiate that. Things that include, for example, lipoic acid versus lipidate. So you can differentiate between the two types of membranes of either gram negative, gram positive. We are trying to do that now with stability as well. So we are trying to incorporate descriptors that account for things like PKPD, susceptibility to protease degradation by different proteolytic enzymes and things like that. So that's something that we're working on. I mean, some of the models that we're running, they're basically plug and play. So we're trying to plug in descriptors that allow us to really, in the end, have a model that autonomously creates a drug-like molecule, right? That accounts for or mitigates issues with stability, PKPD, cytotoxicity, and all those other elements that you really need to make a structure that is a drug. So those are all elements that we're considering.
Thank you. This next question is for Jim. As your model is trained on E. coli compounds, how would you predict activity for other species where efflux and permeability are a really great problem?
So it's a very good question. So our model was trained, as I said, and as the viewer noted, on E. coli data. There, we're now beginning to collect data for other species and for species for which, in fact, permeability and efflux are problems and or features to be addressed. And so the model's predictive capabilities are largely and universally limited to whatever you use to train it. And so in this case, I think some of the interesting features of halicin probably arose more as just an extra benefit and wasn't specific to the training data in any meaningful way. But the questioner goes to a very good point. I think the potential for these platforms are immense, provided we can get to the right data sets to train appropriate models to ask the right question. And we're trying to move this now towards specifically the permeability question.
Thank you. This next question is for Henriette or Fredrik on QSAR models. Is it easy to incorporate 3D structural information rather than 2D only to build such models? And what are the pros and cons for using 3D versus 2D structural information?
So there are methods to do 3D QSAR models. For example, field-based QSAR is one method, and there are tools for this in some of the commercial software suites as well to like calculate these descriptors. It's not typically done because it does introduce an array of various problems. You need to determine a conformation to use for your molecule, which introduces a lot of complexity into your model. But for certain questions where the 3D structure is of importance, obviously they may be the only way to capture the information you're after, but it's not commonly used, I would say.
Okay, thank you very much. Next question for Cesar. In the evolutionary tree, do you see a pattern in terms of short peptides from certain taxa being more effective against medically important bacteria compared to peptides from another taxa?
Yeah, that's a great question. We actually don't have the answer to that. We've, more recently we've been focusing on the human body, and a high percentage of the encrypted peptides that we found hidden within human proteins, they seem to have very good antimicrobial properties, including against clinically relevant pathogens. But we don't have experimental data for throughout the evolutionary tree. In the case of the HTPs in particular, we know that they're conserved throughout evolution. We know that they're conserved through the tree of life, and we know based on data that is available in databases, publicly available databases, we know that a lot of them have antimicrobial properties. But I'm not sure anybody has done that in-depth, sort of an in-depth analysis to really compare that in a standardized fashion. I mean, it's difficult because there are a lot of different peptides that you could choose from.
Thank you. So this is for Jim. Been asked if you could provide a bit more information about the mechanism of action of halicin, and have you tested this molecule on any nitramidazole resistant strains?
We haven't tested against the resistant strains, as noted. We had tested it against more commonly resistant strains across other classes. Here again, the work that John did, he was looking at RNA-seq data that indicated different pathways that were being up and down regulated, and pointed to the significant role for pH. And what we were able to do was run experiments. We could alter the pH in the media, and found, for example, the potency dropped significantly for halicin as pH went up. we similarly found is we could modify PMF and pH with different chemical compounds that we could modulate the potency of halicin, and again pointed to this fairly large scale membrane effect. And it would point to almost biocide type activity for halicin, and yet we found that it had very, very low tox profiles for at least the systemic doses that we used in the mouse models.
Thank you. So this is a question for either Henriette or Fredrik. Which free software do you recommend to perform free energy calculations?
Oh, that is a very difficult question. I am not sure personally that there is any free software out there for free energy calculations. That's something that's been quite recently developed and is only, as far as I know, available in commercial software. I don't know whether Fredrik has any suggestions, but
not that I know of.
It tends to also be expensive in terms of just the computing power. So you tend to need sort of quite a large machine and a lot of data. So yeah, on a budget, it's a difficult thing to do, free energy calculations.
Just could you use GROMAX to do free energy calculations? Perhaps?
Yeah, you could do.
Yeah, and I guess there are a number of tools, and GROMAX and also some other quantum mechanics based tools that are free that you could use to do calculation. But I don't think there are any tool that offers, to my knowledge, there's no tool that offers like a pre-built workflow that makes this easy to do.
Yeah.
Thank you. So I'm not sure who this is a question for, I guess any of you. How do you translate chemical structures to be used for machine learning input? So I think what they're asking is, when you've got your chemical structure, what do you do with it at the beginning of the process?
So I'll start off. It really depends on the application, but a very common means would be to convert it to a SMILE string, which would be a linear sequence of symbols, numbers, that can then be appropriately processed by the machine learning system. Similarly, one can do the same of when you're looking at a large scale output or database applied, is then also convert the predicted molecules to SMILE strings that enable you, for example, to enrich for different substructures that are being predicted by the model.
Yes, a lot of traditional methods also use fingerprints. So you calculate a fingerprint for using a method for your molecule, and then you can use that as input for methods that require like a numerical input. So if you want to train a random forest or an SVM or something more old-fashioned, perhaps these days.
Thank you. And this question really, I think, is getting at the holy grail and something that everyone is aiming for, but knowing the mechanisms of resistance to antibiotics, of which there are very many, could an antibiotic be designed in one single molecule that has every factor that could inhibit resistance? In other words, can we genuinely get a molecule that's got great activity, not toxic, all the rest of it, and is not susceptible to generating resistant mutants?
I mean, I think that's a great question. I think it would be very difficult. I mean, for all of us that work with microbes, we know that they have an incredible ability to adapt. They duplicate in a timescale of minutes. So they're really the ultimate survivors. And so I do believe that eventually bacteria, for example, will develop some mechanism to overcome whatever antibiotic you're putting into. But having said that, we are working on that concept. We are trying to find sequences that repress or make it very difficult for bacteria to develop resistance mechanisms, and we're trying to create new molecules that are enriched in those sequences to see if we can select for anti-resistance sequences. So we are trying to explore that a little bit, although I do believe that eventually bacteria will win. But even if we can reduce the time that it takes for these organisms to develop resistance, I think that will buy us a lot of time and suffering of people that get infections. Another approach would be to combine them with antibiotics, which we've seen potentiating effects with the molecules that we've developed in the lab with conventional classes of antibiotics, therefore reducing the dose that you need to kill the bugs on both sides, and so that also decreases the likelihood of resistance development. So that's another approach.
Thank you. So really have a question now for you, Cesar, and you, Jim, because I think Fredrik outlined the hardware resources that are needed. But the question now is, what hardware resources do you use to screen in the algorithms that you use? And are these very specialist and really only available in high-income countries?
I mean, so I'll start. So we are using an in-house cluster, and we also obtained supercomputer time, for which typically you need to apply. You basically apply for a grant to get time on the supercomputer, and then you either get awarded or not. But if you get supercomputer time, then it's great because it allows you to do a lot of these high-end models, or run these high-end models. So it's not really suitable, I would say, for lower income or developing countries, at least in principle, unfortunately.
So I would amplify most of what Cesar said, and we have similar experience. We largely use clusters and occasionally get access to supercomputers. I would think on low resource settings, it would be valuable to see of collaborating with those in high resource settings, given that the interweb allows us to connect and share. And so, these data, machine learning approaches are data hungry, and thus take a good amount of computing power, both to be generated and then to apply for meaningful screening. But I think it's a nice space for collaboration in that you can access an interface with the clusters or a supercomputer from just standard computing packages. And so I think it's something for us as a community to think about.
Thank you. Well, that's a really good point to end on, that we should all be thinking of working together. And I'd like to just thank everybody today for their really excellent presentations and the responses to such wide-ranging questions. We do have a few that we haven't had time for, and I'm really sorry to those that have asked them that we have run out of time, but we will do our best to answer these in writing for you. So thank you, Jim, Cesar, Henriette, and Fredrik. So I'd like to just move on to the classic, isn't it? I now can't move on. Right. Just to remind everybody about our next webinar, again, as part of World Antibiotic Awareness Week. It's on Friday this week, and it's in collaboration with the World Health Organization. If you wish to join this, do register at the link that is shown below here. And we do hope that you'll come to this. It's on saving children's lives, treating neonatal sepsis. So thank you once again for joining us. It's been an outstanding webinar, and thank you everyone for your excellent questions and our speakers. Goodbye.